Point questions. Okay. So question 1, in simple diffusion, do individual molecules always go down their concentration gradient? Why or why not? Now the answer here was no. In simple diffusion, we have a net flux according to the concentration gradient. But if we look at the actual individual molecules themselves, because of the thermal motion that they're exhibiting, we do not see that they always move in one direction. They will move one way or the other, but the net flux is what we talk about when we talk about the movement down the concentration gradient. Okay, so this is a bit of a tricky question. If we're looking at the total particles, they're moving according to the concentration gradient from high concentration to low concentration. But if we're looking at the individual particles themselves, they're kind of going everywhere, right? Some are going left to right, some are going right to left. We have one-way movement, but the net flux is what is moving according to the concentration gradient. Question 2. Which properties of transported molecules affect membrane permeability in simple diffusion? Which properties of a membrane affect the permeability? So just to kind of backtrack here, when we're talking about simple diffusion, this is a particle that can move straight through the phospholipid bilayer. And so properties that can do that, or molecules that have properties that can do that, are things like oxygen, things like carbon dioxide. So they've got to be small, they've got to be uncharged, they've got to be non-polar in order to get across that membrane by simple diffusion, okay? So small particles, uncharged particles, non-polar particles, or hydrophobic particles that play nicely with fats will go through that membrane. Which properties of a membrane affect the permeability? So this is about the membrane and not the particle. The properties of the membrane are going to be thin. So we said that a membrane that is thinner, obviously less stuff to get through, is going to have an increased permeability. The membrane should also be large enough, so there needs to be a large enough surface area exposed to increase the permeability. And then obviously the general nature of the phospholipid bilayer, which is that it's non-polar. So non-polar, larger exposure in terms of the surface area, and then a thinner membrane in terms of the amount of stuff for particles to get through. So those are some of the features of the particle and of the membrane that would lend for increased permeability. Question 3, for a substance crossing a tissue by simple diffusion, which of the following, choose all that apply, cause the permeability to increase? So the first one here is increased tissue thickness. That is going to decrease the permeability, so that will not be selected, right? If we've got a thicker membrane, more stuff to get through again. Decreased size of diffusing molecules. So if we've got a smaller particle, we would definitely increase the likelihood of that being moved through the membrane. So that would be selected. The third option here is decreased lipid solubility of diffusing molecules. So if the particle or the molecule is not lipid soluble, it is not going to be going through that membrane as easily. So that is going to actually decrease the permeability. So we would not select that option. If we decrease the lipid solubility, we decrease the particle's likelihood to get through that phospholipid bilayer. And then the last option is increase the temperature. Absolutely. If we increase the temperature, again, we increase the thermal motion of particles, and they're more likely to move in general, let alone move across the membrane from side to side. So increasing the temperature, while we said that it's not as applicable in our physiology because our temperature is pretty set, in terms of physiologic fact, increasing the temperature would certainly increase the permeability for simple diffusion. So the choices that we would select here are decreasing the size of the diffusing molecule and increasing the temperature that would cause an increased permeability. Okay. All right, I'll pause for a couple seconds and see if any questions came up from those. Otherwise, we're going to move on to objective 3. Okay. So objective 3 is to compare the movement of molecules across the plasma membrane by carriers through channels. So we'll talk about some of the mechanisms other than simple diffusion by which we can get particles and molecules across the membrane. So the first is facilitated diffusion, and keyword facilitated here. So this type of movement needs help. It needs a carrier. This is still passive transport, however, so we don't need energy. This is actually energy liberating because the particles are moving, again, according to their concentration gradient. But here, because the particle cannot go through the membrane because it's not permeable to the membrane, we need some type of carrier or protein embedded in the membrane through which this particle can move. So this is where we have those transmembrane proteins that we talked about. These are some of the proteins that are embedded in the phospholipid bilayer. And they have specific binding sites that are unique for specific particles. We'll talk about an example with glucose in a minute. And so we've got binding occurring on one side of the membrane. This is the open phase side of this transporter or this carrier. It binds, and then the protein or the transporter goes through a configurational change where it changes its shape, and it exposes the particle to the other side of the membrane, whereby it can simply just exit and it will go through the membrane. So we've got a binding on one end, a configurational change of the carrier, which exposes the other surface to the intracellular environment, and then that particle can be kind of just spit out on the other side, and then the membrane can resume its shape. So this is what we mean by facilitated diffusion. While we're still moving according to a general concentration gradient from high to low, because it cannot go immediately through that phospholipid bilayer, these are larger particles. These are glucose, for example, as we'll see, which is kind of bulky and is not permeable to the phospholipid bilayer. Because of that, it needs a carrier, a protein, a helper, a facilitator, if you will, to get through that membrane. And so what we'll see here is that the rate of transport in facilitated diffusion is limited because we have a fixed number of carriers in the membrane. We don't see that indefinite, linear, directly proportional relationship the way that we saw for simple diffusion. So if I backtrack here a couple of slides back, we talked about this completely linear relationship, directly proportional, which was indefinite. As long as we had a concentration gradient, as long as we increased the concentration gradient, we then increase the net flux or the net movement of particles. Because we have a limited resource back here, so here in the facilitated diffusion, because we have a limited resource, we don't see that completely, indefinitely linear relationship. When we max out the number of carriers, the concentration gradient can still increase, but the net flux is going to remain the same. Okay. So let's look at an example here of facilitated diffusion. This is looking at the GLUT transporter 4, sometimes called the GLUT 4 transporter. It is a protein embedded in the membrane. It is made on rough endoplasmic reticula, which are basically the ribosomes that stud the ER. This is where we make proteins that are made for export out of the cell. It's packaged in vesicles. And so it goes to the membrane and embeds itself in the membrane. Now, when we ingest glucose, this rises or spikes our insulin levels. And insulin is the hormone that is responsible for upregulating or inserting more GLUT4 transporters into the membrane of cells so that we can appropriately uptake that glucose that's in our bloodstream. In things like diabetes mellitus, there's a lack of insulin, which simply means that we've got less GLUT4 transporters in the membrane. And so this is why we have more of that glucose sticking around in the blood and less of it actually being uptaken into the cells where it can be utilized through cellular respiration. So this is what the problem is. This is why diabetes mellitus is particularly problematic, because if we don't have insulin, there's no signal to insert more of these transporters in the membrane of cells so that we can take the glucose out of the blood into the cell and use it to make ATP. So the GLUT4 transporter is how we uptake glucose as an example of one of those transmembrane carriers. Here, the second type of movement is diffusion through channels. So this is quite similar to facilitated diffusion. We're still moving passively, still moving according to a concentration gradient, but here we're not moving through a transporter, we're moving through a channel. This is also a transmembrane protein, but obviously it's shaped a little bit differently and it behaves a little bit differently in that it's simply a passageway or a pore or a channel or like a tunnel or column that's in the membrane. So it's not going to have a configurational change. It's not going to flip-flop the open face the way that the transmembrane carriers did for facilitated diffusion. Here, this is simply just a passageway that exists for those particles to travel through. This is also substance specific, so we don't have water moving through ion channels. We don't have ions moving through water channels. Specific molecules or particles are going to move through specific channels. And we'll talk about some of the examples. So examples of channels can be aquaporins. Aquaporins specifically transport water. Remember, water is polar. It's not going to go through that lipid bilayer, so it's not going to be permeable. The membrane is not going to be permeable to water. So to get water into or out of the cell, we need an aquaporin channel, which is specific to movement of water. Other types of channels are ion channels. Now we have different examples of ion channels. We've got leak channels that simply move ions according to their concentration gradient in or out. We've got gated channels, and within gated channels, we can have different types of gated channels as well. They can be voltage gated, where they're open and close at a specific membrane potential. They can also be ligand gated, where something has to bind to them in order for them to open or close. So different types of gated channels. And then we also have bidirectional channels, which we'll talk about some examples as we move along as well, but simply allow the bidirectional movement of ions or particles. Leak channels only allow one direction of movement. They can be in or out, but it's going to be either in or out, if that makes sense. Some of the factors that affect movement through ion channels are similar to facilitated diffusion. We have a specific rate for each channel. So a channel can only move a certain number of particles through, and then it can no longer move any faster. The second limiting factor here is the number of channels in the membrane, similar to facilitated diffusion. If we exhaust the number, we put as many ion channels in the membrane as we can, obviously we can no longer increase the rate because we've maxed out the number of carriers. And then obviously if we max out the weight of each carrier, we then obviously cannot go faster than that. And then the last factor is basically whether or not the channel is opened or closed. That's going to be an obvious factor. If we've got a closed channel, we can't increase the weight and vice versa. Okay, any questions on those two forms of passive transport? So I want us to keep the big picture in mind here. Passive transport is moving spontaneously downhill. It's liberating or releasing energy. And active transport is going to be the opposite. Among passive transport, we talked about simple diffusion, where particles can go right through that membrane. They don't need any help, any carriers or anything like that. The second type was facilitated diffusion, where they require a transporter or carrier, which has a bit of a configurational change to help that particle move. And then the third type of passive transport is carriers. So carriers are just passageways in the membrane that can facilitate the movement through from one side to the other. So let's look at some of the checkpoint questions here, and then we'll keep moving along. So what is the difference between a carrier and a channel versus how are they similar? So we kind of just talked about this. A carrier and a channel are different because the carrier is actually carrying the particle through the membrane. If you kind of think about the word there, it's facilitating the action. It's actually doing something to help the particle get through the membrane, whereas a channel is just kind of sitting there. It's standing there. It's just a passageway that is allowing the particle to move through. And that's where we get the word facilitated, in that the carrier is actually facilitating or helping or shuttling, so to speak, the particle across. So that's how they're different. How they're similar. They are similar in that they both move according to the concentration gradient. So they're going to be a form of passive movement, downhill movement from high concentration to low concentration. The only thing is that these particles need some extra help because they can't go through that phospholipid bilayer. Question 2, as the concentration of a substance increases, the flux of molecules transported by carriers increases at first, but eventually levels off. And the question goes on to ask why. So this is because when we have movement of particles or movement of molecules through a carrier, we are limited by the number of carriers in the membrane. So at first, when we start moving this particle, we can increase the weight to some degree, but as we max out the number of carriers in the membrane, we can no longer increase the weight beyond how fast those carriers are moving and how many carriers we have in the membrane. So at that point, the graph will then flatten out or level off because we've exhausted our carriers, which is the limited resource. Question 3 asks, when an uncharged substance such as glucose enters a cell by facilitated diffusion, does it go up or down its concentration gradient? And I'm really happy this question's here because I really want to make the point that facilitated diffusion is still a form of passive transport. Although it's not movement immediately through the membrane the way that simple diffusion is, it still is moving according to the concentration gradient. So for example, glucose moves through a facilitated carrier or a transporter in the membrane. It's still going to move from a high concentration on one side, which is in the bloodstream where we've got a lot of glucose, to a lower concentration on the other side, which is on the other side of the cell membrane. So it's still moving from high to low according to the concentration gradient. Okay, I'll pause again and see if there are any questions on those checkpoint questions. And then we'll move on to objective 3 here. Okay, so objective 3 is really talking about active and passive transport. We're going to distinguish between particles that move passively through the membrane and then how we can affect the weight of those movement of particles. So active transport is a whole nother ballgame. This is going uphill. This is non-spontaneous, and just to really break down what the word spontaneous means here, it cannot happen on its own. It needs energy first. Passive transport can happen spontaneously without any energy input, and so it can happen irrespective of any other factor, as long as we have that concentration gradient. Because active transport is moving in the opposite direction, it needs help, right? It needs a booster, it needs energy in order to bring about this type of uphill movement. And so for that energy, it's also going to require a pump of some sort, which is a membrane protein, similar to our facilitated carriers. It's embedded in the membrane, so it's a transmembrane protein. But this pump is going to help the molecule move across the membrane by utilizing energy in order to help this movement. So it's an uphill movement, a non-spontaneous movement. If we think about the images here on the right, if we've got equal concentrations on both sides, we have an active transporter in the membrane that's separating these two sides. We're going to see movement of particles from one side to the other. But at the bottom here, when they get to steady state, we're going to see a higher concentration on side B than what we see on side A. So active transporters can move particles against their concentration gradient. That's what we're really trying to say here. They're not necessarily moving with the concentration gradient, but they can move particles from low concentration to high concentration, which is going against their natural spontaneous movement. And that requires energy. So one type of transmembrane protein here is called the sodium-potassium pump. We're going to talk a lot about the sodium-potassium pump. It can harness energy. It has specific binding sites. So it has 3 binding sites on one side for sodium, and then it's got 2 binding sites on the other side for potassium. And the pump itself has the ability to hydrolyze ATP, use that phosphate. So we talked about the amount of energy that's stored in that third phosphate. So it can grab that third phosphate off of ATP and take that energy to move these particles against their concentration gradient. Now, these pumps can exhibit saturation, meaning that when we've maxed out the number of particles being moved, then we max out the movement, this type of active transport. We max out the movement of these particles. So let's move on here to talk about the two types of active transport. There's primary active transport, which uses ATP directly. And there's secondary active transport, which kind of uses the energy that's liberated from another molecule moving according to its gradient. So in primary active transport, the energy to move molecules or particles against their concentration gradient comes from the hydrolysis of ATP. So that example that I just talked about previously there, we've got this pump which can bind our molecule or ion, in this case in question, and it'll bind those sodiums on one side, it'll hydrolyze ATP to get the energy for this process, and then it'll have a configurational change with that ATP energy, and it will shuttle those ions to the other side of the membrane. Alternatively, it'll bind to potassium on the other leaflet of this pump. It will have another configurational change whereby it hydrolyzes ATP again, and then it'll spit those potassium on the opposite end of the membrane. So this ATP pump can move sodium and potassium in opposite directions against their concentration gradients by simply hydrolyzing ATP to move sodium in one direction and hydrolyzing ATP to move potassium in the opposite direction. So that's primary active transport. We use the energy by from hydrolyzing ATP to move those ions against their concentration gradient. In secondary active transport, we're going to use the energy that's liberated from another ion that's moving down its concentration gradient. So another ion or particle that is moving according to its own concentration gradient. Remember, when we talk about passive transport, we said that passive transport is energy liberating. So if I'm moving sodium from high concentration outside the cell to low concentration inside the cell, that is going to liberate energy as it's moving according to the concentration gradient. And that is indicated here by the green arrow. So this green arrow means that energy is being liberated as sodium moves passively down its concentration gradient. Now, with these secondary active transport pumps in the membrane, we can borrow or steal or grab the energy that's being liberated as sodium moves according to its gradient, and we can take that energy and move another ion against its concentration gradient. This is what we call secondary active transport. So diffusion results from previous active transport of the ion from one side of the membrane to the other. An example of secondary active transport is the sodium-linked glucose pump. So this is the example that we were looking at. We take sodium, we move it passively according to its concentration gradient from high to low, and we use that energy to move glucose against its concentration gradient from an area of low concentration to an area of high concentration. This is also referred to as co-transport because both particles, both molecules are moving in the same direction, and they're liberating energy. One is liberating energy for the other one to move. Another example here is the sodium-linked proton pump. This one is sometimes called counter-transport simply because the particles are moving in opposite direction, but the same principle applies. We're moving sodium according to its concentration gradient. That liberates energy, reflected by the green arrow here, and that energy can be used by this pump to move hydrogen ions against their concentration gradient, which is indicated by the red arrow here. Again, this is referred to as counter because the particles are moving in the opposite direction. Here is a really nice summary table. I've created a similar table in that review video that I did. It's not as extensive, but you really want to look at all of these forms of transport together and be able to compare them in terms of the energy requirement, in terms of uphill versus downhill, and then in terms of the actual transporter. So whether it's using an ion or a carrier or a pump in the case of active transport and so on. So a really nice study table for you to kind of synthesize everything that we've talked about in terms of transport. And then in this final slide, I really want to highlight the importance of the sodium-potassium pump and the leak channels that we talked about. So the leak channels being those ion channels in the membrane moving passively, the pump being an active transporter. All of these components are crucial to establishing the intracellular and extracellular concentration of ions. So the reason that we have high sodium concentration outside the cell, high potassium concentration inside the cell is because all of these components that are on the cell membrane, the sodium potassium pump on one end and the leak channels on the other end. So that sodium can leak into the cell according to its gradient and then be pumped out of the cell against its gradient. And so this helps to establish and maintain our relative ion concentration, not just sodium and potassium, but all of our ions. So chloride, calcium, and even the relative concentration of our amino acids and so on. These are maintained and established by the presence of these pumps and carriers and channels that live on the membrane of our cells.